Hello dear students, today's lab will be about culturing of bacteria. According as we all know that bacteria can grow on solid media, liquid media. The process that we are going to do today is the growing bacteria on solid media. We have three types of agar here: nutrient agar, MacConkey agar, and blood agar. And all the requirement, we have the blood bottles culture, Bunsen burner, we have the inoculated loop, urine sample. Everything is ready to do the procedure. Before we get to the culturing of bacteria, we are going to introduce these culture medias. We have the MacConkey agar, which is a selective and differential medium which allows only gram-negative bacteria to grow and inhibit the gram-positive. That is why it's selective. It only allows gram-negative bacteria to grow. And also differentiated. What do we mean by differentiated? It's a fermenter and non-fermenter. There are some bacteria that have the ability to ferment lactose, like E. coli. When it ferments lactose, the color of colonies will be pink. And non-fermenter, if the bacteria doesn't ferment lactose, the color of colony will be colorless. And then we have the blood agar. Blood agar, which is a fastidious blood. Fastidious means the organism that requires specific environmental condition and special nutrient agar. And it's red blood cell lysis. It determines the hemolysis. There will be three types: alpha, beta, and gamma. The alpha, which is a partial hemolysis, and beta, complete hemolysis, gamma, non-hemolysis. And then we have the nutrient agar. This nutrient agar is non-selective and non-fastidious agar. We mean non-selective, it's quite contrary to MacConkey agar. Nutrient agar is non-selective, which means all the bacteria can grow on this agar, which is a general nutrient agar for growth. And then we have these blood culturing bottles. As we said before we culture blood, we need to make sure that there is bacteria present inside the blood. So the process, before we culture it, we are going to put blood into these bottles. We have the aerobic and anaerobic bottles. Aerobic, which means the bacteria need oxygen, and some bacteria doesn't require oxygen, which is anaerobic. And we put them into a automated analyzer, which back alert for 5 to 6 days, because sometimes in blood there could be fungi exist or bacteria. When we get a positive alert that bacteria is present inside the blood, then we are going to culture it. And now we are going to culture blood on blood agar. Now the blood, we can culture it on blood agar and nutrient agar, and also MacConkey agar. We're going to culture this one. We're going to take we're going to culture this blood, which we take it from the bottle on plate. We're going to place one drop on here, and then the streaking technique will be the interrupted, which I explained on the lecture. First, we are going to sterilize the lobe. When we sterilize the loop, we are going to cool it down, and then again, for each step we are going to sterilize the inoculated loop and let it dry. There are 4 steps involved in this interrupted streaking. And we are going to put it upside down, as well as for the urine sample. Urine sample is easy. We can use this kind of loop, say plastic loop. The urine culture is easier than blood culture. We're only going to put it right here and then streak it. Now we have the blood culture and urine culture ready. After completing the culturing them on agar, we're going to put them in a incubator at 37 degree for 24 hours, and we're going to put them upside down, and that's all. Thank you.